Lesson number 3 in the homeostasis series is going to focus on the first portion of the nephron. So here we have our nephron. We've already labelled, we know the main regions of it from left to right, and we're going to focus on this one. Everything that goes on between the Bowman's capsule, which is this bit, and the glomerulus and associated arterioles here. Do remember in section it has a cellular layer around each side of both these. Let's make it a bit bigger, and this is indeed what it looks like, and we're going to start naming the bits and pieces of it. So we have blood arrives in this arteriole called afferent, A for arrive. The tangle of it we've already named is glomerulus. Blood then leaves the glomerulus and the Bowman's capsule via a blood vessel called the efferent, or E for exit, A for arrive. It's important to be able to recall the differences between these two arterioles. So the afferent arteriole has a greater diameter than the efferent arteriole. In other words, per unit time, more blood can arrive than can get away. The consequence of that is a raised hydrostatic pressure. That's pressure due to fluid, so a raised hydrostatic pressure. Now be prepared to see these diagrams switched around anyway. So they ask you in this one, which is afferent and efferent, give you reasons. You have to look at the diameter of them. It's not which one's above and which one's below. So it turns out in this diagram, that's the efferent, and that is the afferent. Why? The greater diameter of the afferent. They're arriving, so blood is coming in via that one and out via that one. Now, as stated earlier, that increased blood flow in compared to out generates a very high hydrostatic pressure, an increased hydrostatic pressure. That's the driving force for molecules. If they're small enough, they're filtered out of the plasma into this part of the nephron. It will be then called glomerular filtrate or just filtrate. You have to know what makes up the barrier. There are three parts. One, there are the endothelial cells. So if it's small enough, it can get between the endothelial cells. Then it has to pass through a membrane that holds all the endothelial cells together in a capillary called the basement membrane. That is the most selective. That is the most important bit of it. It then has to go through the wall of the inner wall of the Bowman's capsule, and they're made up of special cells called podocytes. So there are three parts to the barrier. One, the endothelial cells. Two, the basement membrane, most selective. And three, podocytes. Here we have a diagram showing these podocytes. So here's a capillary, so the endothelial cells and basement membrane there, and this is an inner wall cell of the Bowman's capsule called the podocytes. And here's an electron micrograph of it, and you see it has literally finger-like projections that actually hold on to the capillary, and anything that comes out needs to pass through those little fine gaps, also known as filtration slits. In another section it looks like this. So this is inside the blood vessel here. Anything needs to come out needs to go through the endothelial cells, through the basement membrane, but fit in between these finger-like processes of the podocyte.